As you can see, the title of my talk, as Garrett said, is Your Brain a Quantum Orchestra as opposed to a computer? And we'll get to that. And I'm kind of giving away the plot here in the quantum orchestra. This is a neuron, and most hierarchical models of the brain and consciousness would go from the neuron up to networks and networks of networks and so on and so forth. But the point is, we have to go down, inward, deeper, faster. And we now know with experimental data that inside the neuron, in the microtubules, we have operations in kilohertz, megahertz, gigahertz, terahertz, petahertz, and faster, and via the Penrose mechanism, presumably all the way to the Planck scale. But I'm going to talk about only within the brain, primarily today. So most view the brain as a complex computer of simple neurons. Neuron firing equals 1 bit. This is obviously an artificial neural network, but that's pretty much what our theories of consciousness are based on, with consciousness theorized to emerge from higher order network effects. This is an insult to neurons. It only considers the membrane and the synapse, nothing inside, only at Hertz frequencies, up to, say, EEG frequencies, purely algorithmic. There's no real room there for consciousness, free will, and so forth, I'm arguing. So what's the alternative? Well, to illustrate that point, single cell paramecium, if we're treating a neuron as a 1 or a 0, which is what most theories do, single cell paramecium can swim, learn, avoid predators, find food and mates, and have sex. There's 2 paramecium engaged in sex. No synapses, no networks. They use microtubules and cilia to sense and navigate. And being means question mark consciousness. We don't know if they're conscious. If they are, most likely during sex because they're absolutely still, and that would reward procreation, of course. And we'll come back to that point. The paramecium shown there has these, the cilia are these hair-like extensions that come out of this paramecium that are both oars and sensors. And they sense and move by these cilia are made of 9 doublets of microtubules linked by dynein motor protein arms, which contract to cause purposeful bending and locomotion. And it was proposed by Adam in 1974 that signals propagate along the microtubules, tell this one to contract, then this one, so you get coordinated, purposeful motion, which is how the paramecium moves. And microtubules are also found in neurons, as illustrated in this cover from Neuroscience. The long, straight, these things here, and they're interlinked by microtubule-associated proteins. And this is an axon because they're all parallel and uninterrupted. Now, what does that have to do with the cilia and the dynein? If we look inside a neuron, and here's a neuron with the axon going here, and in the dendrite, we're looking inside and at synaptic plasticity. So synapses anywhere down here need supplies, need enzymes, need precursors and so forth that are synthesized here, and they're transported along the neuron by along microtubules serving as tracks with the dynein, the same motor protein that was in that's used in paramecium to bend to actually carry material. In this case, the dynein's carrying it back to the cell body, and the kinesin, the other motor protein, is carrying it the other way. Now, how do they know where to get off? Which synapse needs replenishing for synaptic plasticity? And that's the tau protein, which is a microtubule-associated protein, which is placed at specific locations on the lattice of the microtubules. And this signals the motor proteins, they're kind of traffic signals, to deliver their cargo to particular synapses. So this is learning. This is memory right here. This is the placement of the tau directing synaptic plasticity. Now, when this falls apart, when the microtubules, when the tau falls off the microtubules, it forms neurofibrillary tangles. The microtubules destabilize and disassemble. You lose neuronal volume, you lose brain volume, you lose memory, you lose cognition. And that's called Alzheimer's disease. Now, there was a paper I read in 1989. Many of you weren't alive then, probably. And it was about Alzheimer's hypothesis, microtubules, a key to Alzheimer's disease. 1989, Matsuyama and Jarvik from UCLA. It's an excellent paper. I recommend it. It'll explain how the problem is caused. It makes some mention of the amyloid plaques, but they don't really cause the cognitive dysfunction. It's due to the loss of microtubules and the tau. So I'm going to raise two questions at this point. Why are we spending billions and billions on toxic and ineffective anti-amyloid drugs for Alzheimer's instead of treating microtubule stability and resonance? And that's a whole nother story. But for this purpose of this talk, do microtubules process and encode information? And I first published on that in 1982 in the Journal of Theoretical Biology with my colleague Rich Watt, an engineer who knew about computer matrices and so forth. So is memory encoded in microtubules? We don't really know where memory is encoded. If we say it's in synaptic plasticity proteins, these last hours to days and memories last lifetimes. And the most likely site for memory encoding is in microtubules for one mechanism is this calcium calmodulin kinase 2, CAMK2, which is activated by calcium influx into the neuron. These kinases pop up, and these hexagonal little creatures, they're enzymes, bind perfectly to the hexagonal lattice of the microtubules and kind of phosphorylate up to 6 tubulins at a time. And this work was done, led by Travis Craddock, who is here and works at Nova Southeastern. And we published this back in 2012, I guess. So this is a potential mechanism for memory. And the memory encoding capacity of microtubules is enormous because each tubulin can be in one of 30 different genetic or post-translational states, and you have about a billion per neuron. So a billion raised to the 10 to the 30th power, that's a lot of possible memory states in one neuron. Can they process information? Back in the 80s, there were these cellular automata, the game of life, the simplest form of computer. And basically, you start with a orthogonal grid, and each square can be dead or alive. Dead in this case means alive. And with very simple rules, if you have 0, 1, or 4 live neighbors in the previous generation, you die because there's either not enough or too much overcrowding, not enough sustenance or too much overcrowding. 2 and 3, you stay alive. Those very simple rules give you gliders that move through, and if you make a large enough cellular automata, you can solve partial differential equations and do all kinds of stuff. So my colleague Steen Rasmussen and I and some others modeled microtubules as cellular automata, which is different because it's a skewed hexagonal lattice with Fibonacci cylindrical geometry. And black and white in this case represent opposite dipole orientations, which oscillate with each time generation. So you need a clocking mechanism. You need some kind of coherent clocking mechanism. And for that, we applied Froelich coherence. And Froelich was a biophysicist in the 60s and 70s who suggested that coupled dipoles in nonpolar regions in proteins, geometric arrays, and lattices would oscillate coherently in terahertz, 10 to the 12th hertz, gigahertz, and or megahertz, condensing to common modes, pumped by ambient the heat bath. So it takes biological heat into a lattice and pumps coherent modes, something like a laser. So that was Froelich coherence. It was a theory. Do microtubules have collective Froelich coherence suitable for clocking frequencies? And here you can see the microtubules inside an axon and inside the dendrite where they're interrupted in mixed polarity, and the other one I showed you before. So are there oscillations? Yes, the answer is yes. Anirban Bandyopadhyay, working at the National Institute of Material Sciences in Tsukuba, Japan, has studied microtubules, and in a series of papers about 10 years ago, looking at 3 different scales. So here we have a neural network. Here's a neuron, a couple other neurons with some nanoprobes coming in. The thick bars are nanoprobes. And here's one microtubule with 10 nanoprobes, and here's rows of tubulin with 4 nanoprobes. So 3 different scales. And in all of them, if you apply a voltage to a microtubule, it's an insulator. You don't get current flow. However, if you apply AC, alternating, and sweep the frequency, you will find certain frequencies that the microtubule becomes highly conductive, almost superconductor, ballistic conductance it's called. And this occurs in the same pattern repeating every 3 orders of magnitude. So if we start at the smallest, here you can see terahertz, gigahertz, megahertz, and then we move up to gigahertz, megahertz, kilohertz, and then megahertz, kilohertz, and hertz. And we see the same pattern every 3 orders of magnitude, which is a triplet of triplets. So you can sort of see it here, but you can see it over there from an aerial view. And so we see 3 peaks, and each peak has 3 peaks. This is seen every 3 orders of magnitude, over 12 orders of magnitude. So microtubules do have coherent self-similar resonance patterns. The triplet of triplets repeat every 3 orders in hertz, kilohertz, megahertz, gigahertz, and terahertz. And there's Anirban, who's done some amazing stuff. He also showed that you can measure megahertz from the scalp in the EEG. So look on the screen, you can see a triplet there. When he takes it off, it goes away. He puts it back, you can see the triplet on the screen. He takes it off, it goes away. So this is between 6 and 26 megahertz, which is where it's found in microtubules. So we see the same triplet. We don't quite see the triplet of triplets, but we see triplets from the scalp. And this, I think, is going to be a revolutionary new aspect of EEG in its 100-year anniversary, to find out that the hertz that we've been measuring all these years are only the slow end of an iceberg. I'm mixing metaphors here, of much, much faster stuff going on inside the neuron. So another study that Anirban's group did in neural networks found megahertz and gigahertz excitations, not just passive resonances, but excitations in neuronal networks, and they regulate axonal firings. In other words, they're functional. So here's a neuron, a part of a neural net, impaled between 2 of these chips that have these nanoprobes that are dielectric resonance detectors that can measure megahertz, gigahertz, high frequency stuff. So if the probes go through the neuron, as we see here, they detect megahertz and gigahertz. So it's detected only from the neuron, revealing their shape. So there's no megahertz and gigahertz out in between the neurons. And if you look at a microscope of this, you can see the neuron. This is in kilohertz, and it's showing ionic fluxes, which are outside the neuron. And the megahertz and the gigahertz is inside the neuron that we see there. So this is mapping out the neurons by where the megahertz and gigahertz is coming from. Now, they also found that the dendritic somatic megahertz and gigahertz correlated with axonal firings on the same and different neurons, more so than membrane potentials. So the deeper, faster activities were overriding the membrane potentials, and including the integrated membrane potential, and forming their own little circuits. And in the picture there, which was the cover of the lead article in Journal of Physiology, you can see that you see the neurons, but then you see different little networks that are actually due to what's going on at a deeper, faster level that don't necessarily follow the anatomy. So there's a whole nother level of activity. He calls them filamentary circuits because they're coming from the cytoskeletal filaments, including microtubules. Now, this explains something called representational drift. There's this problem in neuroscience where if you see a group of neurons that have certain memories stored, and you can tell that they're there, you go back later, and those same memories have moved a little bit to a different set of neurons nearby. There is representational drift, and so it's hard to understand how that could happen from synaptic plasticity memory, but it's easy to understand if it's happening at the microtubule level in a kind of a holographic memory, and it's just shifting